we're going to talk about phage display. The first thing that we want you to know is that all the 3D images that appear in this video were made by ourselves, along with some other pictures in 2D that appear without a mark. So, what is this phage display technique about? Well, this innovative technique was used for the study of the interactions between proteins and other molecules on a large scale, taking advantage of certain type of bacteriophages, like the one we have here. This method was first invented by George Smith in 1985, for which he won the 2018 Nobel Prize in Chemistry. Well, actually, he had to share it with Francis Arnold and Gregory Winter, therefore getting only one-fourth of the prize. But look at him, he's still happy, anyway. But the real importance of this breakthrough is actually the applications that it has, like the development of antibodies, cancer treatment, or even COVID-related applications. This will be described further on. But what are really the bases? Well, this is all possible thanks to the fact that we can introduce foreign DNA into the phage capsid genes in order to synthesize a fusion protein. This fusion protein will be displayed on the phage capsid and it will include the peptide that corresponds to the inserted DNA. Once we have the displayed peptides in the phages, we can study their interactions with other molecules by affinity selection. Now, after this little summary, I suggest you prepare yourselves to start with the detailed explanation. From now on, we'll use this 2D simplification of the phage to make things clearer. The first step is to display a peptide on the virus capsid, as the name says. The most common vehicles to do so are E. coli filamentous bacteriophages, such as M13, like the one we have here. The filamentous phages capsid contains a coat protein named P3 that is located just in the tip. But how do we introduce the DNA into that gene? Well, the vector used to introduce the DNA is called a phagemid, that is a plasmid that contains an origin of replication from a phage, so it can be replicated as a plasmid in bacteria and can also be packaged in viral particles. By doing this, we manage to display the peptides as fusion proteins with the P3 capsid proteins.
Being more specific, a random DNA sequence encoding for a small peptide is inserted into the P3 gene. When this protein is synthesized, the peptide is also synthesized along with it, forming the fusion protein, represented in red in the picture. This process is repeated, introducing different peptide sequences into different phages, and then infecting a bacterial culture in order to obtain millions of phages expressing different peptides. This is called phage library. In his experiments, George Smith used a library of 250 million different phages, all mixed together. The purpose of making this gigantic library is to find a random peptide that can bind to a certain receptor. This is achieved by affinity selection, making it possible to find among all the phages which ones are able to bind to the receptors. First, these receptors are immobilized by attachment to a petri dish. The library is added. Therefore, if any of the peptides from our phages has a high affinity for the immobilized receptor, the phage will bind to it. Then, the dish is washed to remove the ones that haven't been attached to the receptors. Now, by elution in acid, the bond between them is weakened to release the wanted phages. Only a couple of them will be obtained, so they are amplified by infecting a bacterial culture. Finally, the peptides from the selected virions are individually sequenced by sequencing the DNA that was inserted into the P3 gene originally. These selected peptides have then a very similar amino acid sequence to the domain from the original ligand of the receptor that is inserted, with a high enough affinity so that they are able to bind to each other.
So now, thanks to Javi and Anna's explanation, we already know what phage display is, but we still don't know the applications of it. So I'm just going to discuss this topic, and just for you to know, there are tons of applications of phage display, but I'm just going to focus on 3 of them. The first application has to do with enzyme specificity and inhibitors. In this case, I've chosen 2 examples of it. The first one is the one that we use phage display to bind to allosteric or the active site of the enzyme. In this way, it acts as an activator as an inhibitor. In this case, the enzyme will act as the selector, as explained before, and the random peptide bound to the phage is the one that is going to either activate or inactivate it. In the second example, we can see that we can inhibit protease activity in enzyme thanks to phage display. But how? Well, we can seek the peptide that will bind to the protease site thanks to phage display. After finding it, we will amplify that selected phage, and then we will add the peptides to the enzyme. They will join the protease site, impeding the protease activity, and then obviously making them resistant to cleavage.
The second application has to do with a really important topic, that is cancer. So as we all know, chemotherapy and radiation are two real important methods to fight against cancer, but they also have significant side effects. Because of this, we should consider also immunotherapies. These therapies will provoke an antigen-specific immune response by delivering tumor antigens and adjuvants. And just for you to know, adjuvants are substances that will bind to these antigens, making the immune response even more effective. So as we know, if the immune response is even more effective, will mean that these therapies, the immunotherapies, will be much more specific. But how do you apply this to phage display? Well, as phages are strangers to the human immune system, cells will phagocytize them. If we have applied phage display, as we know, these phages will have peptides attached to them. So thanks to the MHC 1 or 2, the peptide will be shown attached to the complex in the extracellular space. The peptide will be recognized by T cells, provoking an immune response that will kill these tumor cells that provoke cancer. The third application has to do with in vivo techniques. In this method, we use phage display to inject phage with peptides into live organism, for example, a mouse. After letting the phage circulate through the mouse blood, we collect the tissues and sequence the phage. We should consider that this process is normally produced several times so we can identify the peptides that affect a specific organ. Apart from these three applications, we also have a fourth one. It is the most important one, and it has to do with antibodies, but it's going to be discussed by my classmate Bea.
One of the main applications of the phage display technique is the production of antibodies. And I thought it was suitable, taking into account our current situation, our current context, to approach it to COVID-19 treatment. And I know what you might be thinking, phage display to treat COVID-19 infections? Aren't you basically saying to attack a virus with another virus? Yes, that's exactly what I'm saying. But don't you worry, phage are not dangerous to humans. To explain how phage display could be a way to neutralize COVID-19, it's important to clarify that the spike proteins of the virus were found to be the major domain responsible for mediating the membrane fusion, being the transmembrane glycoprotein ACE2 the receptor for the virus. This receptor is found in the membranes of human cells, more specifically in the lungs, kidneys, blood vessels, and intestine. Therefore, the binding between the spike proteins and the receptor is key to COVID infection. Therefore, generating specific antibodies that recognize the spike proteins of the virus could indeed stop COVID-19 infections. It is important to point out that we'll only produce the viral part of the antibody, which is the binding region of the antigen and where the neutralization will take place. To produce the antibodies, we will infect a chicken with the spike proteins of COVID-19 to extract its antibodies, more specifically in immunoglobulin Y. In brief, for phage display technology, first, the lymphocytes containing the specific antibodies are isolated from the host animal that has been immunized. Then, the messenger RNA encoding for the antibodies is retrotranscribed into complementary DNA. The viral regions of the antibody cDNA are amplified by PCR and then ligated with each other into one fragment. The amplified cDNA is inserted then into a phageme. The phage is used for E. coli transformation, and then the bacteria are infected by helper phages as to induce the expression of the phage. The result is that the bacteria expresses and releases phage particles containing the vector DNA, showing the engineered antibody protein on their surfaces. This way, we've created phages that act as antibodies that will bind to the spike proteins of the COVID-19 virus, and this binding will stop the interaction between the ACE2 receptor and the spike proteins of COVID-19.